Hi, my name is Benjamin Brakel, and I'm an undergraduate student in Dr. Sheila Singh's lab at McMaster University. My research here focuses on glioblastoma, or GBM, the most aggressive primary adult brain tumor which currently has no cure. Patients usually present with large tumors, as shown here in the MRI, which remit after standard of care, including surgical resection and intensive chemoradiotherapy. However, the tumor always recurs due to a therapy-resistant population of glioblastoma stem cells. Understanding and targeting this dynamic, heterogeneous population of cells is essential to inhibiting this recurrence and improving patient outcomes. We recently performed functional genome-wide CRISPR knockout screens on several patient-derived glioblastoma stem cells in our lab and discovered that these cells rely on a protein known as roundabout guidance receptor 1, or ROBO1, involved in axonal guidance early on in development. As ROBO1 is involved in development, it's not normally present on normal, healthy brain cells. However, using flow cytometry, we discovered that it is highly expressed on the surface of our glioblastoma stem cells. This is shown on the left, both in primary and recurrent GBM lines, which all show extensive ROBO1 presence. The table on the right further shows surface expression of ROBO1 on many of our other cell lines, and again, it's highly present across our GBM cells, as well as on other brain cancers, including childhood medulloblastoma, as well as brain metastasis, while still low on normal human astrocytes. To target this vulnerability, we designed and engineered a panel of camelid-derived single-domain antibodies against human ROBO1. We then used these antibodies' binding regions and incorporated them into the extracellular binding domain of chimeric antigen receptor encoding DNA. This DNA was then transduced into human-derived T cells, creating ROBO1 targeting CAR T cells in hopes of training these immune cells to eliminate ROBO1 expressing glioblastoma stem cells. Our antibodies bound to ROBO1 in a comparable fashion to commercially available antibodies at low nanomolar concentrations. Additionally, we made a direct comparison between our antibodies, all of which showed high affinity and specificity for ROBO1, to select that with the highest affinity. We then orthotopically engrafted GBM cells into immunodeficient mice and administered intracranial treatments of this antibody or of control antibody. While mice treated with the control antibody consistently showed tumor growth and engraftment as expected, 75% of mice treated with our ROBO1 antibody were tumor-free after treatment, an example of which is shown here on the right through immunohistochemistry. After using these antibody binding regions in developing CAR T cells, preliminary studies of several CAR T cells with these differing binders showed that binders 4 and 5 were the best candidates, and we proceeded to in vitro assays with 2 variations of each of these CAR T cells. When co-cultured with GBM cells, these CAR T cells demonstrated specific and potent killing, as shown on the left, in green and in blue, which was not shown with cells not expressing ROBO1, as shown in red. This also shows that the binder 5 CAR T cells were clearly the most effective, and so we proceeded with these in our next assays. These CAR T cells showed high levels of activation, as shown by CD25 and CD69 expression on the right, after exposure to GBM cells. We are now conducting further in vitro validation of this therapy, including assays of cytokine release, CAR T cell proliferation, and antigen density of ROBO1 in relation to CAR T cell killing, and will soon begin in vivo studies as well, where we are confident it will reduce tumor burden and prolong mouse survival. To finish, I'd like to thank all the organizations that have supported this work, as well as Dr. Sheila Singh and the rest of the Singh Lab. Hi, my name is Benjamin Brakel, and I'm an undergraduate student in the lab of Dr. Sheila Singh at McMaster University. My research focuses on glioblastoma, which is an aggressive brain tumor with no cure. Patients usually present with large tumors, as shown here in the MRI on the left, which disappear after standard treatment, as shown in the middle, but inevitably recur due to a population of glioblastoma stem cells, which don't die from these therapies. The standard of care hasn't changed in over a decade, but we've recently discovered that these stem cells resistant to this therapy rely on a protein called ROBO1. As ROBO1 is involved in development, it's not normally present in normal brain cells. However, here we can clearly see that it's highly expressed on the surface of our glioblastoma stem cells, as shown by the percent of cells expressing it in red. The table on the right further shows this percentage for many of our other cell lines, and again, ROBO1 is highly present across our GBM cells, as well as other brain cancers, including childhood medulloblastoma, as well as brain metastasis. To target and eliminate these cells, we took immune cells called T cells from human blood and gave them the DNA that we engineered that encodes a receptor. And this receptor recognizes ROBO1, and when it does bind to ROBO1, activates these T cells to induce killing. In a way, this trains these killer cells to find and eliminate GBM cells that have ROBO1 on their surface. On the left, we see that these trained immune cells greatly kill our tumor cells, as shown in blue and in green, while they don't kill cells not expressing ROBO1, as shown in red. These CAR T cells are also highly activated, as shown on the right, being within the top right quadrant, which indicates high levels of activation markers. We are now conducting further in vitro validation of this therapy, and will soon begin in vivo studies in mouse models where we hope it will reduce tumor burden, as well as prolong survival of these mouse. Thank you.